Hello, my name is Alexandra Crook. I'm from the University of Nebraska-Lincoln, and I'll be presenting today on the combining two analytical techniques with chemometric analysis to characterize wine by vineyard, region, and vintage. I'm a bioanalytical chemist by training, so this was a really fun project. It was highly collaborative. We worked with two other universities to perform this research. So just a little bit overview, a year in the life of wine. Most of us are probably familiar at some level with this, at least as samplers. But wine goes through several stages from growth, which is going to determine your region, soil type, and climate. Mashing is where you are adding the, you mash up the grapes and you can remove the skins or leave the skins on. This is where you'll typically introduce starch to sugar. Combination with yeast through fermentation. And then aging, it's usually left to age, sometimes in oak barrels, depending on the vineyard, and then bottling. And at each stage, you introduce new characteristics to the wine, and these are going to be heavily dependent on how the wine is produced. But our research was specifically focused on growth. We wanted to eliminate as many of these variables as possible. So we were specifically looking at grape wines of the Pinot Noir clone 667. And this clone was planted at 15 different vineyards along the Pacific coast of the United States. You can see here I have colored the regions we're looking at, all the way from Southern California in the red and orange up to the Sonoma Valley in the blues. And then we have two vineyards up in Oregon that we're looking at the Willamette Valley. And these grapes were all harvested at similar sugar concentrations to try and eliminate any variables between the harvest. And they were all harvested between August 13th to September 15th in 2015. And then we had a second vintage that was harvested between the 25th of August to September 21st in 2016. And all of the wines were developed and harvested by the Runbaum Lab at the University of California, Davis. So this is kind of our experimental design. We took these 15 different vineyards, 2 different vintages, and 8 different regions, or AVA regions. And we performed, at the top we performed NMR metabolomics on these. So looking at that chemical snapshot of the 15 different wines. And on the bottom, I'm showing that we have our chemometric, or our differential sensing analysis. This is looking at the phenolic compounds, and it's a color sense assay in which we have peptides that are bound to a chemometric peptide, and that peptide is replaced through the phenolic group, and then we get a color change. And that's how we are looking at those phenolic compounds, and those kind of mimic the taste of the wine. So we're looking at a very untargeted technique through our NMR spectroscopy, and a more targeted technique through this differential sensing technique. And we performed both multivariate and univariate analysis on both of these independently, and then combine the data sets to see, can we see an improvement in our classification. So here I'm just breaking down the different regions we have. Again, we have multiple regions looking at the Santa Maria Valley and the Santa Rita Hills that are going to be in our Southern California, Arroyo Seco, and the Sonoma Coast, Sonoma Carnosa, Sonoma RRV, which is the Russian River Valley, and then also the Anderson Valley and Willamette Valley up in Oregon. So we're trying to get a good breadth of the Pacific coast. And we had 2 different vintage years, 2015 and 2016. And we did 8 analytical replicates for each group. So the first step in our analysis was looking at multivariate analysis. We use an MBA Pack software tool that can take NMR data. This is specifically designed for NMR data at this point. You can see in the image that I'm showing is we can take it all the way from raw data through our models and validation. And so that's what we use for the NMR data. We can also take the differential sensing assay data through a CV data and import it and run it through our models as well, starting at steps 7 and 8. And then finally, what we did was we combined the 2 data sets in a multi-block analysis to give equal weight to each technique. So we were able to run both of them through it in this software package as well. For our feature analysis, we use MetabolAnalyst, which is a common tool in our lab as an online chemometric tool that was used to analyze the features. Random forest was used to test the classification strength of the data based on the vineyard, region, and vintage. We did 500 trees with 7 predictors. And we use the out of bag error, or the inverse of the out of bag error, as our classification score. We also did rock curves. Rock curve analysis was used to test the classification strength as well, but we were also more importantly interested in using this as a way to look at what features are being chosen time and time again. Since we have two different techniques, we wanted to see are NMR features being utilized to classify these wines, or are the differential sensing metrics being used as well, to kind of gauge how our techniques are stacking up next to each other in this multi-block situation. So the first thing that I'm going to be looking at is our Vineyard classification. So we're looking at those 15 different Pinot Noir wines that were derived from the same clone. And our goal was to utilize the two distinct analytic techniques to improve our classification of the vineyard specific characteristics. And I'm showing here the 15 different vineyards we had along our 8 different regions. So here I have 3 different PCA models. So PCA was used to carry out the NMR and assay data separately. As you can see, our NMR data showed a lot more separation. That ended up being something that we expected, just because our NMR data, we were able to get a lot more variables. We ended up breaking the data up into 214 bins. So we have a lot more data to work with than our assay, which we had 27 distinct measurements. So we weren't surprised when our NMR data was able to give us a wider breadth, but we're able to see some distinction that we were happy with in our data. So something that we kind of just by eye looked at was that you can see, since we have these color coded by region, we see even like our southern coast here, we see these red, orange, and yellows that are clustered here, and then we see them clustered as well kind of in the background here. And we were able to see when we put them in together in the multi-block that we were able to see that same trend carry through. So we see some of our characteristics carrying through in that multi-block analysis for the 2015 Vineyard as well. We performed this again on the 2016 Vineyard data, and we saw similarities as well. Our assay data did not perform as well as the NMR data as expected, but we were really the NMR data was very much an untargeted look, whereas our assay data was a very targeted look. We were looking for very specific details. So we wanted to be able to pair these together to see how they complemented each other. And we were able to see similarities that we saw in the 2015 as well. One of the ones that we really liked was that we were able to see that clustering again of those Southern California wines, and some of our greens too, denoting that our Sonoma Coast Valley wines were clustering, especially in the NMR, and our Russian River Valley up here, although they do see some here as well. So we did see some distinct separation between those vineyards that we were hoping to see. The assay data was a little bit more clustered, which is not unsurprising when you think of Pinot Noir wines and how they taste. But our multi-block data, we did see some separation as well that we were very happy with in terms of the classification. And we also did show here, down here, we have our dendrograms that I showed in the previous slide as well, looking at the Mahalanobis distances as p-values. So in looking then at the feature analysis, we first looked at random forests, as I'm showing here. We have the NMR data, the assay data, and then the combined. So we ran all of our data through separately and then as a multi-block to see which features were we able to improve our classification. And some of the things I'll note is that at some points, especially like in our PMSA, we were able to see that both saw high classification, but then when we combined them, we saw improved classification. And in some sections, especially in the Ross, NMR outperformed the assay, and we saw that in the combined data set as well. But in like Bloomfield, we saw similar results across the board. But overall, this is just a simple average with standard deviation. We were able to see a marked improvement across both the 2015 and the 2016 vintages, which we were very happy to see. We then were very interested in looking at the rock curves, what features were coming out of this, what features were unique to the vintages themselves. So we used the area under the curve as a mark of the classification strength. And then we also looked at, you'll see this in the next one, we marked how many features were being utilized and what percentage of the data set that incorporated. And that was really important to us because as I said, we have 214 bins in our NMR. So even if 20 of our NMR bins show up and 5 of our differential sensing assay showed up, we wanted to make sure that we were showing that our differential sensing assay was contributing significantly. So some of the things that was interesting, what we saw, especially in Cloud Landing, in this line, in the NMR for the 2015 vintage, we only saw 20% contribution of our NMR data and 80% was from the assay. But then we saw that flip into 2016 and we saw 80% coming from the NMR and 20% coming from that assay data, just showing that these both data sets are giving us unique values and unique information to help our classification. So then we also were looking at something like ROS that showed consistency across the NMR data was much more valuable in separating and classifying this data overall. So we next moved into the region classification, and this was where we knew we were going to run into a little bit more difficulty because we're looking at we're trying to combine a lot more data into specific areas. So as you can see here, this is the NMR and assay data as PCAs, and you can see even like with the pink group here, which is our Willamette Valley, you can see they're clearly distinct in their vineyards, are much more distinct than their vintage, but we do see some separation in the vintage. I really like that the right here, our Santa Maria Valley, it does cluster quite nicely, but we can still see those very distinct groups in there. The assay data struggled a bit to see the regions over the vineyards, and that may be due to the fact that this is a much more targeted technique. So we did this as well with the 2016 data, and we again see that we can see some separation, especially our Santa Maria. We can see very clear groupings of our vineyards over our regions. And you can see that the 2016 assay data, which I've unfortunately mislabeled as NMR over here, this is our assay data, struggled again to see that those distinct regions. But what we wanted to also see is even if they couldn't multivariately see this distinction, could our assay and our NMR show distinction in the features that they were selecting? And so what we're able to see is that the assay and the NMR were performing quite well in their random forest techniques, and we can see here that we see lower values in the individual techniques than when we combine them. And we sometimes see it again, like we saw previously, is that sometimes the NMR performed better than the assay, and that carried over into the combined analysis as well. And this is just showing that our combined assays gave us a marked improvement over the individual techniques. We then also showing down here again the rock curve analysis showing our area under the curve and the distinct features that we have. So this is just showing that we have, again, that sometimes our assay data was able to outperform the NMR data, especially in specific regions. So we see here in the Santa Rita Hills that the assay data in 2015 gave about 36%, and the NMR data gave about 64%. But then in 2016, we saw a flip of that and an 80% contribution of the assay data to the NMR data, which is something that we were highly encouraged by in looking at this, the feature selection. The last thing that we did was kind of look at kind of the what features were being pulled out across in both of the vintage years. So the NMR features usage for all of the rock curves was plotted using NMR bins of a ppm value of about 0.1 ppm. And we have the vineyard up top and the region down below. I've color coded them by year. So the 2015 data is colored in blue, whereas the 2016 data is colored in red. And specifically, I also plotted the differential sensing assays for the rock curve data as well. And so something that we were really interested in looking at, especially because we have NMR as a technique, is can we identify any specific features, any of those specific metabolites that are popping through that are being featured time and time again in our rock curves. And we were able to see that we saw some branching amino acids over here. We also saw TCA and sugar metabolites, which were expected based on our wine. And those are very highly showing up in our analysis. So those sugar contents, even though they were harvested at similar sugar concentrations, we're still seeing differentiation based on their vineyard and their region, which was very exciting for us. So yeah, we saw things like isobutyl alcohol, malic acid, phenyl alcohols, mannitol, and then a lot of sugars, ethyl acetates, fructose, and then also some amino acids like tyrosine, and then citric acid is also part of the TCA and your sugars as well. We also did a comparison of the vintage year as well. And as you can tell from the data I've been showing you is that our vintage years were very separate. They showed very distinct multivariate and feature analysis. And so we were able to demonstrate through ROC curve that we had an 88.5% area under the curve modeling 25 features, 16% contribution from assay, and 24% from NMR. It'd be interesting to do this for more years. We had a limited sample set from 2015 to 2016, but it'd be interesting to see if this trend continues across years as well. So overall, that was the fingerprint of our study, was the combination of NMR and this colorimetric sensing. Fingerprint improves wine classification, and we can see distinct features that are coming out of both techniques. So overall, I'd just like to say a special thanks to our collaborators, Dr. Eric Ansel and Dr. Diana Gamora Olveras, and her students at the Freshman Research Institute at the University of Texas at Austin. I also don't have a picture here, but I would like to also thank Dr. Ron Runenbaum. He was the one that produced all of our wine for us and was very instrumental in helping us to understand kind of what these features tell us about the wines and the regions that we're looking at. And I also like to thank my lab as well. So overall, thank you.
Questions? So can you say exactly how both the colorimetric assay and the NMR, and you concluded, I think you said that they are together providing a better resolution of classification. It seems like the NMR is really doing a great job. I don't really understand if the colorimetric assay adds something or.
Yeah, so we were able to see that we did see, it was a difficult situation because the NMR had so much more information available than the colorimetric data, but we wanted to use that targeted technique because the chemical signature that we're seeing, we can't really attribute that to what you taste in a wine. We can say that there's more malic acid or there's more isobutyl alcohol, but like we really wanted to look at, because a lot of Eric Ansel's work has been looking at this sensory mimic and saying, what do we see any taste differences? And saying, can we identify those taste differences based on the region of growth? So tracking all the way to that beginning statement. And so what we were able to see is that we can get high classification by NMR alone, but we wanted to say if adding in a second technique can improve it all. And I think that we did see some marked improvement overall. It was a really fun study, and I think that the combination of a lot of techniques have been used, and LCMS has also been used, and they've shown some really cool results.
So it would probably be if you could just add all of it, NMR and colorimetric assay, some sort of feature set, machine learning problem, or dimensional relativity problem, maybe that.
I'm sorry, can you repeat that?
Take all the data, forget about classification, this assay does this, that. Just put everything in one big feature set. And then you do your PCA or whatever dimensionality reduction you want to do and see whether that improves classification of the code.
So we put them all into one data set for the random forest and the rock curves.
Okay, all right, oh yeah.
Yeah, so that's when we combine them, we put them all into one data set. But for the multivariate analysis, we did give them equal weight. So we took them as two separate data sets and put them into the model together with equal weight.
All right, so thank you very much.
So I kind of have a question related to what he was asking. So remember, there are like for the 2 years, the assay and NMR data set is kind of flipped, like the ratio that contributed to the rock curve. I'm just wondering if you have a chance to look at like the common features for the 2 years and like the unique features, and if there's any takeaways, if you look at them, like if there's specific chemical signature that you found or something like that.
Yeah, so that's what we were doing here is we were looking at what features came up in the rock curves time and time again. We did look at the overall what was coming up. We didn't take it that step further and look at if we could see the very unique features to each wine. We found that they tended to show up time and time again, but we didn't look into a comparison if they were increasing or decreasing compared one to the other. That would be definitely another step to go and say, can we give distinction to each one? if we did that analysis, I would like to get more years, more vintages to say, can we see a marked increase in this vintage or in this vineyard across maybe 4 or 5 years? That would be really interesting. We are limited to 2 years for this data set, which is why we didn't go that far and say like, Willamette Valley has an increase in malic acid, because we only have 2 years. It would kind of be probably improper to say that with the limited data set, but we did say overall, can we say from year to year, do we see differences and do we see marked increases in the features that we're selecting?
Yeah, I just thought it would be really cool if we were able to make the connection between like maybe the metadata of grapes and like the features, you know.
Yeah, it'd be definitely something more to explore.
Yeah.
There's a lot of information to explore in this data set. It would be really fun to keep working on it.
Yeah, thanks.
Yeah.
Thank you, very, very nice work. And my question is, I was expecting, or maybe it's a feeling that, so when you merge, we work with both data sets, you would get much better classification. It was not, so it was a little bit better. So I was wondering how much redundancy you have between these two data sets. So if maybe a canonical correlation will tell you how much redundancy. And then my question is, if you did any kind of feature selection before feeding the random forest, or you just put all the features available?
We put all of the features available. The only thing we removed is that in the NMR data set, we're doing a multi-suppression technique to minimize the ethanol signal. So we did remove our ethanol regions from the data, just because they are 10, 20, 30 fold increased intensity compared to the other features in our data set. So we did remove the ethanol for the NMR data set. Otherwise, we threw everything in to our analysis. And to your first question, we had played around with merging both data sets together and seeing, can we see differences in the regions and the vintages? And we struggled for a long time to try and get both data sets to give us a uniform analysis, and that's still something that we couldn't achieve at the end. We ended up moving towards looking at the vintages as a separate entity because they were quite unique, which would be, again, interesting if we could have more vintages and say, is there a pattern that's overarching? Because I think that with the 2 years, we couldn't see an overarching pattern among them. And there's a lot of information that Ron Runenbaum's lab has looked at and saying year-to-year comparisons, just because he has a lot more knowledge on the viticulture work.